Hello, everybody, and welcome to the SCHOTT Pharmaversity podcast. My name is Neus Ferre, and I'm going to be your host today. The topic today is Antibody and Radionuclide Drug Conjugates, Advanced Vial Solutions for Cutting-Edge Cancer Treatments. To talk about these so-called ADCs and RDCs, I have with me Diana Löber. And if Diana is here, you may imagine the connection between today's topic and SCHOTT Pharma. And probably you all guess, yes, it's the glass vials. Diana is Global Product Manager at SCHOTT Pharma, and she's responsible for the vials. Diana leads and coordinates development around glass vials. So, as you can imagine, she's our vial champion. Hi, Diana. Nice to have you here today.
Hello, Neos. Thanks for the introduction. Nice to be here again.
Great. So I would say, Diana, let's start. So when we were preparing this podcast, Diana, and we talked to each other, you already mentioned to me how important this specific topic is to you and how much it could change cancer therapy. So would you mind giving us a brief insight into your thoughts on this topic?
Yes, definitely. I mean, besides heart diseases, cancer is the second leading cause for death in our society, with a strong tendency to grow within the upcoming decades. This is a disease that affects us all, sooner or later, directly or indirectly. So my own grandmother died back then in her mid-30s, leaving behind her husband and her daughter, so my mother. And right now, I know 3 women of my circle who are currently under treatment for breast cancer or just finished the therapy. And all are in their 30s or even 20s. So cancer is a devastating disease.
Yeah, unfortunately, I totally agree. At the same time, Diana, however, my perception is that recently there have been many great advancements in early diagnostic and also in treatment, right?
Yeah, sure. There have been huge advancements in the last decades. And the increasing survival rate looks good. But nevertheless, these treatments take their toll on the patients. Chemotherapy often leads to lifelong burdens, since certain substances are toxic to the heart, the lung, muscles, and so on. Not mentioning the great risk for infertility. And radiation can also lead to cardiovascular or gastrointestinal diseases or result in skin tumors as long-term effects. This is mainly due to the fact that these therapies are still too random. And what do I mean with this? These therapies not only kill the bad guys, the cancer cells, but there are huge collateral damages as well. Healthy tissue is also being killed.
So kind of, yeah, surviving comes at a high cost.
Yes, and I even know people who told me that they wouldn't do it again. And we didn't even talk about the psychological burden. So there is room for improvement.
Mm. So today we talk about this antibody and radionuclide conjugates, ADCs, RDCs. How can these, let's say, newer drugs help, and what makes them so interesting?
So if you allow me, I will take a few steps back. I think it is very helpful to understand some basic mechanisms of our immune system to understand the operating principle of these new approaches. And therefore, I would like to mention antibodies. This is something that I guess everybody mentions from time to time when having a cold or during COVID, everyone talked about that as well. But I think that not everyone knows what actually stands behind that term. Yeah.
Yeah, I guess you're right, and I would be one of them. So we'll use this term loosely, and probably only half of us, in best case, have an accurate idea what actually it means.
Yeah, that's my impression too. So often it is referred to the lock and key principle. Yeah, so this is described when it comes to the interplay between antigen and antibody. But for me, this is only one part of it. Actually, the antibody is like a tracking dog, yeah, in our immune system that is specifically trained to identify and possibly deactivate a specific matter. Like there are, for example, tracking dogs that are trained to detect drugs or others to detect dynamite or other tracking dogs. they detect humans, yeah? So each of them has their own field of expertise. And as soon as a tracking dog arrives at its target, it either alerts others or it attacks the suspect by its own. The principles that antibodies typically apply to inactivate viruses are called, for example, neutralization, opsonization, acclimatization, and so on. So I will not bore you with any more details at that point. Important is to understand that antibodies are great when it comes to hunting, to finding the right target and act upon it. And this is an ability that you can perfectly exploit when it comes to cancer treatment. This is exactly what traditional chemo and radiation therapies cannot fulfill, identifying and attacking the cancerous cells only. So the reason why there is such a huge collateral damage.
Mm-hmm. Yeah, I like this metaphor with the tracking dogs. So let's say, as you say in your words, antibodies are these tracking dogs that are specifically trained to find, in the case of the ADCs, the cancer cells. Is that correct?
Yes, and if you know a little bit about working dogs, it takes ages to train them and it costs a huge amount of money. So that's another analogism.
Okay, so now we have the antibody, the tracking dog that can bring, so let's say, or reach the cancer cell. Okay, and what's now? Based on the name and the naming, so there's more than only this antibody.
Yeah, so the second very important term is conjugate. So this means that something is attached to it, is paired with it. In case of a classical ADC, an antibody drug conjugate, the drug that is linked to it is typically a toxin. So simply speaking, an ADC is a locally applied chemotherapy with the huge benefit that if it works properly, it only kills cancer-affected cells.
Yeah, which sounds pretty amazing. Okay, so if that is the explanation for the ADCs, we have the other kind of, let's say, of the family, the second part of the family, are the radionuclide conjugates in the title of the session. How about them? Probably same principle, Diana.
So the addition idea is to use the same mechanism to realize a local constrained radiation therapy. Instead of attaching a toxin, in this case, a radionuclide is attached to it. Goal here is again to only irradiate and therewith kill cancerous cells. And these are then radionuclide conjugates. They're also called RACs or ARCs or RDCs. So there are different abbreviations and not a finally implemented one, to be honest. Yeah. But actually, the idea of using radionuclides for diagnostic reasons, so imaging of the cancer, has been established for a long time. Yeah. But instead of using it now in a diagnostic setting, the idea has been developed further to use it for therapeutic purposes. So not only for identification and visualization of the cancer, but for attacking it. And first, peptides with a covalently bond DOTA by a functional calculator have been used to realize so-called peptide receptor radionuclide therapy. And with the evolution of the therapy, now also antibodies are used to guide the radionuclides towards the cancer cell.
So actually, the main mechanism or the idea would be the same. So to kill the cancer with a toxin or radiation, but now the major difference lies in the fact that it can be executed in a much more targeted way. Is that correct?
Exactly. With the benefit that less, or in the best case, even none, healthy cells are killed. Imagine the potential behind that. It could revolutionize cancer therapy.
Absolutely. So less side effects, less lifelong struggles. Yeah, sounds pretty convincing. So nice to hear that you are such a huge fan and ambassador of the ADCs. You are at the same time the product manager for vials, and now we come to the connection. So what should we understand here, Diana?
Yeah, so as you can imagine, ADCs are very delicate, very sensitive molecules. Just think about our prior sessions, Neos. There we highlighted how sensitive monoclonal antibodies are and that they in many cases require a dedicated primary packaging. Now we are talking about a molecule consisting of 3 parts. So monoclonal antibody plus linker plus radionuclide or toxin, the so-called payload. Often multiple linkers attaching multiple payloads are used to increase the efficiency of the ADC. So makes it even more complicated, let's say, yeah? And this is why in almost all cases, ADCs are being lyophilized, as are, by the way, more than 50% of the monoclonal antibodies as well. Yeah? So I only know of one ADC which is in clinical trial in liquid form at the moment. So we have and will have a huge amount of ADCs being lyophilized, I assume.
Mm-hmm. And what is it actually that these drugs need to be lyophilized? So everyone talks about the stability, but what is actually behind that?
Yeah, so what typically happens is that especially big proteins form aggregates. So you could say that they stick or glue to each other. In some cases, you can see that really by naked eye, so the solution somehow becomes cloudy, and sometimes it is not visible at all. Yeah. So, but the consequence might be that the drug loses its activity, or that it leads to an increased immune response, but also is not desirable. Yeah.
I assume this would be a no-go. Okay, so what is the challenge with the lyophilization step?
It is a very complex process. So it is very difficult to develop the freeze-drying cycle, especially upscaling, from laboratory to commercial is really a challenge. And then when being in commercial, big freeze dryers have capacities sometimes for more than 100,000 vials. Yeah. So can you imagine this? This is huge. Yeah. And the thing is, you put it in, you close the door, and then hopefully when you open it, usually 2 or 3 days later, yeah, you hope everything is fine. Yeah. And if it's not, it is a disaster. And in some cases, so-called fogging occurs. So the naming comes from its appearance. It looks like a fog, yeah, that stays on the walls of the vial above the lyo cake. This impairs inspection, but also really is an issue for the acceptance of the end user, because it looks bad to the one who mixed it so with the liquid, yeah, again, who does the so-called reconstitution. And based on customer requests, we have noticed some risk of fogging when it comes to ADCs.
Mm-hmm. Yeah, and so I can assume that maybe there's something you can propose about it.
Yes, there is. What has been proven is that a hydrophobic inner surface of the container avoids fogging. And therefore you can, for example, use silicone. But if you have a drug of which one vial costs multiple thousands of dollars, you do not want to risk having silicone in your drug product. Yeah. And this is why we offer a product called Top Lyo. It is a vial with a hydrophobic inner surface based on an SiOCH coating. Unique about this is that it is covalently bonded, so to the glass, to the surface, so the strongest bond you can have on molecular basis. And we are able to do this because we have a patented technology, so-called PICVD, plasma impulse chemical vapor deposition. I could now talk an extra hour about this, so therefore I will stop here.
Yes, we can do another session on that. So coming back to the vial, so you can use, let's say, this kind of dedicated vial to improve the lyophilization process.
Yes, to avoid fogging, but also receive a smooth, nice lyo cake. Because an additional pain can be what is called cake disruption. So the cake is cracked, like torn apart. Yeah. In addition, the hydrophobic coating has the advantage of improving residual emptying. So especially for high-value drugs, or if you need a very accurate dosage, this is very beneficial as well. So studies have shown that you can improve the extracted volume by roughly 5%.
Interesting. So this would be regarding the ADCs, and there would be special, let's say, advanced vial called Top Lyo. We've talked a while about the ADCs. let's jump to the other part. So what about the radionuclide conjugates, the RICs or RDCs, where this, instead of a cytotoxic particle, there's a radionuclides attached to the protein or antibody. What about them?
So as mentioned earlier, when it comes to radionuclides, there are actually two applications. So one, diagnostic, second, therapeutic. And typically for the diagnostic indication, where you want to label the cancer to make it visible, technetium is used as an isotope. With the therapeutic indications, there's a tendency to move from gamma emitters towards beta or alpha. Latest data has even shown that alpha emitters don't go that deep as beta or gamma radiation, but are more able to effectively kill the cancer cell. So with regards to DNA damage, beta radiation only broke the second strand in I think 50% of the cases. Alpha has been successful to break 100% of the double strand. The downside is that then multiple doses are necessary. And upside is that they have a longer half-time and are more durable.
Mm-hmm. So this would mean that these drugs stay in the container for a longer period of time.
Yes, exactly. So the diagnostic drugs are usually used within hours after filling because they decay that quickly. With a longer half-time, radionuclides used for therapeutic applications can last for multiple days, even up to a week. But this means that they are exposed longer to a potential interaction with the container they are in. Even that we know about all advantages of glass and its inertness, there is still interaction between the container and its content. There always is a certain level of so-called leachables. And apparently, especially these drugs tend to react with sodium, calcium, aluminum, which are all part of the glass composition. It affects the coupling of the isotope.
Mm-hmm. And how could you help, or can you help to mitigate this tendency, Diana?
Yes, of course we can. So actually we have two products that can help here. So both solutions are vials that have, again, an inner coating. Important to state is that these coatings are applied by the same process as the earlier mentioned top lyo. Yeah. So again, a covalently bond coating. One is called type 1 plus. Yeah. So this is an SiO2 coating. So actually, we take the type 1 borosilicate glass and we apply an inner coating to it that is pure quartz glass.
Mm-hmm. So this one's a kind of word. Why then just use the vials made up of pure quartz right away?
You could do this, but the melting temperatures would be much higher. You easily go from roughly 1,500 to 2,000. And in addition, the forming of the vial, yes, to receive accurate dimensions would be much more challenging. And this is why we do it like this. So we keep the great advantages of type 1 borosilicate glass, but combine it with a coating that is beneficial for certain applications. This is, by the way, why it is called type 1 plus. Yeah, so it took me years to recognize that.
Okay, makes sense. Yeah, keep the base, no, the vial structure, and then just put the coating that you need for every specific application. Very interesting. So, what does this silicon oxide coating now help with? Why having these pure quartz glass inside?
Yeah, certain drugs can be sensitive towards certain ions. As I mentioned earlier, for example, the chelator that links the radionuclide tends to react with sodium, calcium, and aluminum. Yeah, so these are all parts of the composition and therefore leachables of pure borosilicate glass. Yeah, pure quartz glass, on the other hand, provides the best hydrolytic resistance. So, easily speaking, this glass is the most resistant when it comes to leachables, the most inert one. And with the SiO2 coating, a barrier is established that ensures minimum interaction between the drug and the container. So minimum risk for somehow affecting the drug.
So you mentioned a couple of minutes before, there's a second option helping to cope with the leachables. What would be this?
Yes, so there is a second one that can cope or helps with leachables, and this is every care. So again, a covalently bond coating same procedure, same process applied, but in this case, it is an SiO-C8 coating. And this has been specifically designed for drugs in high pH. So let's say starting around pH 7. So it provides a stable, leachable barrier for these applications.
Mm-hmm. Could these coatings you've mentioned also help in the case an ADC would be liquid form?
So as mentioned above, to my knowledge, besides one, all ADCs are lyophilized at the moment. But we know that, of course, our customers are trying to formulate them in liquid. And for other molecules, we have seen in the past that a coating can help to ensure stability in liquid. So it's definitely worth a try. Yeah.
Absolutely. Okay, so now we've talked a lot, Diana. What would be your, let's say, takeaways or kind of brief summary for the audience?
Sure. So I will give you three news. I will give you three. So first,
perfect.
With radionuclide drug conjugates and antibody drug conjugates, we have potential candidates here to change the world of cancer treatment. Yeah, that's the first one. Second, due to their sensitivity, ADCs need to be lyophilized mostly at the moment. If this is the case, we have top lyo to help here. Yeah. And third, last but not least, RDCs or ACs or whatever they are and will be called are in liquid, as might ADCs be in the future as well. Here we have leachable barrier coating. So type 1 plus for low to medium pH and every care for high pH. So yeah, that's it. Why did it take a whole podcast with this?
I'm happy we took the time, and I appreciate this very much. Thanks for the interesting talk, for your explanations, which are, as usual, very easy to follow. So I really appreciate that. I really hope the audience followed us through the challenges and the solutions you propose. Maybe what I take with me is that in case you are developing or plan to develop and get involved with the world of the ADCs and RDCs, pay special attention to the primary packaging, which may contribute to find a joint solution which allows us to reach a lot of patients in the shortest possible time. And remember that, SCHOTT Pharma, so we have the broadest range of dedicated solutions to safely package, let's say, advanced drugs, and in that case, the ADCs and RDCs. I know, as I understand they are really complex structures, so it's of most big importance to pack them safely. Diana, it was a pleasure. All the best in your projects endeavors, and thanks to the audience for hearing. Hear you soon and see you soon.
Thanks, Niels, for having me.
A pleasure. Bye-bye.
Bye-bye.